So shout out to the organizers and the selection community for this incredible opportunity. Before I jump into the science, I just want to acknowledge upfront my support village, mentors, and funders that have allowed me to really season science in my own way. Okay, so my work focuses, a core focus of my work is on mitochondrial dysfunction, or I like to call it cellular engine failure, because engine failure analogy really gets to the big picture. And regardless of where your engine dies, if it's at home or on the highway, you're about to have a bad day, and especially this economy. So the silver lining is that you can take that car to a mechanic and maybe switch that engine out, pay a little bit of money, hopefully not too much, and keep using that same vehicle for a lot longer. And this is the kind of hope for mitochondrial transplantation. It's a means to replace bad engines in cells. And over the last decade, the interest in this research space has really exploded. And to really appreciate the science shenanigans I've been pursuing, I'll have to catch up first on where the field of mitochondrial transplantation is. So what we know so far is that there are a few methods by which cells will donate or procure mitochondria. The first being through making these tunneling nanotubes that physically link the donor and recipient cell. The next is through like encapsulation, where either through like microvesicle derived from cells, or we can actually have the ability to make lipid nanoparticles that will deliver mitochondria to cells synthetically. And cells can also just uptake unmodified extracellular mitochondria from the environment. And so I initially wanted to see whether we could use mitochondrial transplantation to boost immune function in the context of aging, and particularly in T cells. And so just a quick run through towards T cells. So in generally, when you look at a young T cell compared to its age counterpart, the older T cell has a reduced ability to use mitochondrial respiration or OXPHOS, and has high levels of oxidative stress. And that directly contributes to the HT cell's inability to respond to similar stimulus at the same magnitude as the younger counterpart. So they don't display the same markers of activation, they don't produce the same magnitudes of cytokines, and they don't tend to proliferate to the same extent. And the cytokine production tends to be biased towards pro-inflammatory cytokines. And so I thought that myotransplantation would be a simple way, a potentially simple way to augment the activity and function of HT cells, and that we could use centrifugation to introduce healthier mitochondria into T cells. And that's actually what we did, and it was pretty simple. All we had to do was really augment the amount of mitochondria to T cells in suspension to really increase the efficiency of uptake. And after the mitochondrial transplantation, those HT cells had better OXPHOS capacity, it had lower levels of oxidative stress, that then also led to better functional outcomes or improvements. So they had the HT cells after mitochondrial transplantation had better markers of activation, they produced more cytokines, and they proliferated a lot better. And we even did experiments where we take these sort of reprogrammed T cells and then put them in mice and then challenge them with infections. And here I'm showing you some of the data towards the influenza experiments that we did, and we saw that the mice that received the T cells that received mitochondrial transplantation, they fared a lot better than the other groups. And so because it was a very simple procedure and it was like super reproducible, I was like, well, why not just use this for other cell types? And I became interested in just applying this method to things beyond just the immune system, like the cardiovascular space, which I'm currently working in, with the ultimate goal of making mitochondrial transplantation into a scalable therapy for clinical use and clinical applications. But something that became pretty apparent was that beside the centrifugation method, the other available methods of transplantation were not as efficient or scalable. And so I'm showing two other publications describing using the tunneling nanotube method, and they got about a 10% efficiency, and this other method got up to 40%. And so it just seemed that the scaling, the ability to scale mitochondrial transplantation was not feasible. And one way to look at this right now is like in the current space, if you can do a systemic injection of mitochondria into an organism or even an individual, and it kind of acts like, and the mitochondria behave like they're getting on a bus where you kind of hope that they get to the right, stop or destination or organ. And I've been interested in kind of making more of an Uber system where we can actually tell the mitochondria exactly where to go. And to do this, took some liberty from and some inspiration from antibody drug conjugates, or ADCs. So in ADCs, a drug is tethered to an antibody, and the antibody hones the receptor on a cell, and then depending on whatever that drug is, it might have some sort of reaction in the cell. In cancer, that tends to lead to some sort of cell death. And so we figured if if we could add homing antibodies to the outside of mitochondria and sort of make like a antibody mitochondria conjugate or a mitotag, it would lead to better targeting and internalization. And so I came up with a simple protocol where you could culture donor cells, sprinkle a little secret seasoning onto them, and then isolate these sort of like bioengineered modified mitochondria, complex them with antibodies, and it takes less than 30 minutes. As proof of principle, I'm showing you a mitotag that was created to target endothelial cells. So these are cells that you really can't take out the body. So in every red dot that you see here is mitochondria that has been internalized by the endothelial cell. But I know it's not enough just to get them inside the cells. It's really figuring out what happens after internalization. So do the donor mitochondria get eliminated right after uptake? Does it actually force the cell to eliminate the native mitochondrial pool? Is there sort of a coalescence or fusion between the native and donor mitochondria? And ultimately, is there sort of retention value to doing this sort of experiments and therapy? And I don't have all those answers right now because there's ongoing experiments, but I'm going to show you some of the work that we've been doing to sort of get towards those questions, and some of it entails confocal microscopy. And so at first, I'll just show you some of the data, just comparing the mitotag system on the bottom versus the unmodified or naked mitochondria. And you can see visually that a lot more mitochondria get into cells, and they're co-localizing to the cell's native mitochondrial network. And we sort of confirmed this by flow cytometry just to quantify actually how much more mitochondria is getting in. And it's easy enough to scale this to 100% efficiency of uptake, again, by just adjusting the volume of mitochondria. We've done some other co-culture experiments where we take endothelial cells and vascular muscle cells, which are CD31 negative, to show that there's some sort of specificity towards the system or the technology. And so here you're seeing the endothelial cells in gray, the vascular muscle cells in green, and then you see that the mitotags are preferentially being internalized still by the endothelial cells. And again, another confirmative experiment by flow cytometry to show that it's 100% efficiency of uptake into the targeted cell type. And then the final piece of data that I'll show is towards like retention. And so we've been using these row zero cells which lack which they lack mitochondrial DNA, and so they lack functional mitochondria altogether. And because their OXPHOS is broken, they need aerobic glycolysis and uridine supplementation to survive and proliferate. And so we figured that if the delivered mitotags are functional, we could remove the uridine from cell cultures, and the row zero cells would somewhat have a sustained proliferation. And that's kind of what we're seeing right now with the cells, row zero cells cultured in uridine are in white. The ones in gray are the row zero cells without uridine, and then in red are the row zero cells that had uridine removed but were treated with mitotags, and that's a single treatment. And so we see that there's sustained proliferation, suggesting that not only the mitotags are functional, but they're actually providing a benefit towards the cell type of target. And so what I'm really showing you is kind of like the backbone of a large-scale mitochondrial engineering pipeline. So now we can use things like CRISPR or mRNA nanoparticles to manipulate the nuclear genome, and then using either synthetic mitochondrial DNA or use current mitochondrial DNA editors to really modify and then repackage that mitochondrial DNA back into mitochondria, then kind of deliver it back into cells to see how it might augment and influence cell function. And I'm particularly interested in using this to help repair and resolve mitochondrial dysfunction within the immune cardiovascular axis, at least to start. And that means tackling immune aging broadly, looking at T cells as well as innate immune cells like macrophages, chronic inflammation and infections, as well as how they contribute to the chronic cardiovascular diseases. And if you really think about the amount of engine failure that happens in aging, there's too many possibilities for therapeutic options, for therapeutic opportunities, and I can't really do it all by myself. And so I think this is a great collaborative tool that I'm really ready to deploy and share. But just to highlight some of the projects that I've already started, collaborating with, this one with NASA, where we're using the mitotags to combat space radiation to improve the amount of time we can actually travel in space. Everybody wants to go to Mars, but we're not really sure how we're getting there. And actually, if you're in the Philly area, we're presenting on this today and tomorrow. And another project, we're going to dose aged marmosets with immune-specific mitotags to see if we can reduce systemic inflammation, and for how long does that like reduction in inflammation last. And then the last project I'll highlight is funded by the Boris Wellcome Foundation, where it's really allowing me to build a repository and customize mitochondria with sort of some natural properties and some non-natural properties. And with that, I think I hope you got the gist of what I'm aiming to do with this technology. And if you have questions, come find me.